Great, thank you, Jacob. It's been a real pleasure to be at this special program. It's a topic that I'm very fascinated and interested in called the math of intelligence. I don't think we actually understand the math of intelligence enough to have much of anything but a first program on it. But that's great. That means our jobs we'll have jobs forever. So I'm going to talk about some work for the first time in a public setting that I've been sitting on that I'm really excited to share. This is work with many collaborators, some of whom you see here. And the title is From McCulloch-Pitts to Retina. So first show of hands, who here knows what McCulloch-Pitts is? What it even means, person, place, or thing, right? Okay, great, that's fantastic. I'm really excited. So for the rest of you, I'm going to tell you about McCulloch-Pitts. I think it's one of the most exciting discoveries in the history of science. But first, I want to explain a bit about my title. So one of the big inspiration for me in my life has been my dad, also my mom, but particularly in science, my dad. And he has this book with the most amazing title I think I've ever seen. And I had to do something like that because this is such a wonderful title. I mean, From Logos to Trinity, I think it's an amazing title. So here's the outline. It's going to have 3 parts. Now, just to be clear, this is going to be like a storytelling show and tell talk. I think everyone's pretty anxious. It's the end of the day. Tomorrow's a big day, and I just wanted to kind of, let's have a little fun. Let's be playful with the science here. So the first thing I want to do is to go over how mathematical thinking about how brains compute has huge benefits, and that's the McCulloch-Pitts in the title. Just to be clear, the mantra here is, I even wrote it down because I wanted to make sure I didn't forget it. So respect the constraints and properties that biology has to reverse engineer the math of intelligence. I think this is a fundamental idea, and we should see it being carried out in part 1, and I want you to think about that as we move forward. The second part of the talk is a claim, essentially, that biological sensors, in particular retina, is intelligent. I think this is a pretty easy to see claim, but I think some people might disagree with this statement. And then part 3 is how do we understand the constraints and properties of biological systems in order to reverse engineer and create a new approach to signal coding that's inspired by these sensors. Okay. So part 1. So what can mathematics, essentially, brains teach us about engineering and life and so forth? And I really love this quote. So if brains were so simple we could understand them, we would be so simple that we couldn't. Okay? It's like, it's amazing. It's even hard to make this into a mathematical statement because it's recursive. So you can't even formalize this, but we all understand what this means, right? Hopefully the core participants in this program understand this statement and how influential it's been. So let's go first. So what is the property or constraint of biology that started neural networks and computers and all that business? And I would claim it's the spike. Okay, so neurons, they used to think neurons were like sending liquids through tubes through the brain because that was the steam engine at the time, the steam engine idea. But it turned out no. It turns out they're sending spikes, zeros and ones. Here's an action potential. You might have seen this before. And we of course it's not a discrete object, but we say it's a 1 when it reaches the peak there, and that spike travels down and acts into other neurons. Of course, this is one pump, okay, in that system. So it's really not fair to call this spike or not spike what's going on, but because here's the pump. But that's what we're going to do anyway, and this is what McCulloch-Pitts did. So here's a quick outline of the history of from spikes to neural networks. So first there's spikes, like, oh my gosh, spikes, that's great. And then Ramani Kahal, that's actually one person, he looked in the retina and he saw, wow, there's all these structures. It's not just like spikes, it's just these combinatorial structures, graph structures. And this is all before Turing. And then Turing comes around, here's a Turing machine, and invents the computer. That's wonderful. And then now what I'm going to be talking about for the next few minutes is McCulloch-Pitts. So McCulloch-Pitts comes out in 1943, and my claim is that all of this stuff afterwards is the result of McCulloch-Pitts. Okay, so what's the setting? The setting is in the 30s. This was a really big deal. In biology, they didn't have these notions of physics. And the idea from Reshevsky's school was let's let's treat biology like we treat physics problems, right? So what do? So we try to make idealized models of these processes, analogous to the idealized physical processes, and we make assumptions about the essential features. They spike or don't spike, right? We represent the features in mathematical variables, create equations, and then leverage those equations by solving that to make predictions of the mechanisms of the biology. This is a big deal because they weren't doing this before. This is a really big, big progress in the field. And now comes this this paper. So this is the McCulloch-Pitts paper. So there's two people. One is named Warren McCulloch, and there's Walter Pitts. And here's the title: A Logical Calculus of the Ideas, Imminent Nervous Activity. I mean, what a title, right? So what is McCulloch? So McCulloch is a neurophysiologist and a psychiatrist. Walter Pitts is a mathematician. And just a little background. So Walter Pitts was a wunderkind. So when he was 12, as you do, you go to the library and you find Bertrand Russell's Principia Mathematica at 12, and you dust it off and you read it cover to cover, right? And then you even send Russell a letter saying, like, you made a typo on page 212, and so forth. I think he's probably one of the 5 people that read that book completely. So that's Walter Pitts. When he was 15, he was homeless, and then he was plucked, from there to go to Chicago because they recognized he was a genius. Of course, after this letter to Russell. And McCulloch is a neurophysiologist, and he was really interested in in mathematizing the brain, but in the way that sort of a neurophysiologist would do it. And he thought of these spikes as sending psychons. Okay, what's a psychon? It's like an atomic unit of mental effort or mental activity, right? So psychons. He was like psychons, and he teamed up with Walter Pitts, and they made this paper. So what's really fascinating about this paper, other than its title, is here's the 3 citations in the entire, it's a long paper, and 3 citations in the paper. So you'd think there'd be something about neurons or neuroscience or experiments or anything like that. It's 3 logic books, essentially. That's the entire bibliography. Okay, so easy bib tech, okay? Just 3 citations, and they're all logic, okay? Another thing I want to point out is, so does this look like a logic gate to you? This is the first logic gate was ever put down on paper in this paper, and it was not a logic gate for them. Of course, all of their methods could be used to solve logic gates, and they, in fact, created the first system to solve logic gates. But this is the hot-cold illusion. So what's the hot-cold illusion? If I touch something that's hot, well, I go, oh, that's hot, right? Turns out if you touch something that's really cold, you feel like it could be hot, but if you keep your hand there, you realize, oh, it's actually cold. And they were trying to model the hot-cold illusion with these spiking networks. And they didn't care that they were logic gates and they invented computer design, but that's what they wanted to do: solve real problems of behavior. So what is the McCulloch-Pitts network? So it's also it's kind of like a perceptron if you've ever seen it. I just want to impress upon you what it is mathematically, because this is IPAM, right? So we've got to do a little bit of math. So what is it? You have a network of neurons, and again, all simplified. Xi and Xj are 0 or 1. They're spikes or not spiked. Jij is the connection. And here's the dynamics. Okay, so think of it as a blinking in time network. Same at neuron I. What I do is I take the sum, the feed-forward input, so the sum of the linear combination of the neighbors. You check if it's above a local threshold, and then you fire, and otherwise you don't fire. Okay, that's really beautiful. It's so simple, and everyone thinks, well, how could that do anything interesting? Well, it turns out that it's Turing complete. You can make this Turing complete. It was actually just a little footnote in their papers, like, oh, by the way, this is Turing complete. But one thing I want to press upon you here is that when von Neumann saw this work, he said, oh, wow, I can make AND gates, NOT gates, I can make a computer, right? So very shortly after, he had this 80-page a paper called EDVAC. It was the first programmable computer design. I mean, his machine. Von Neumann machines today are the same, and they're his machines, right? So this is the von Neumann. And what I think is really fascinating is that he cites 2 papers in this 80-page description of the computer design, and they're the same paper, and they're McCulloch-Pitts. Okay? He's not a very good speller, but that's okay.
I have to ask, out of curiosity, in what sense was this the first and Ada Lovelace's stuff was not the first, a century earlier?
I don't know if she had this exact dynamics.
No, not that that's dynamics, but in terms of gates and being programmable and all that, she did.
I don't think Lovelace did, but Shannon's master thesis.
Shannon certainly. Yeah.
Shannon's master thesis, which I think directly inspired him, was.
So Shannon came before this?
Yeah, Shannon was 36. It's about the same, 37, I think.
Well,
Yeah.
And Lovelace, of course, was a whole, whatever.
She didn't have logical gates.
But she had programmability.
She did program, but the connection to logic was Shannon.
Okay, yeah.
I think the history of computation is it's an unbelievable story, going all the way back, ancient times, Babbage, Lovelace, all these things. So, okay, so what did this paper do? So I urge you to check out this survey paper here by Piccinini. What it did, just a couple things, very few small things. It invented finite automata. It inspired logic gate design, which they actually solved how to design these gates, even though they didn't care. First use of computation for the mind-body problem, and first modern computational theory of mind and brain, which look kind of the same, but they're different. It was really like the idea that stimulus comes in, neurons interact in a dynamics, and the idea of baboon comes to you like somehow as a psychon or something, right? So that's what's going on. Another thing is I don't want to belabor this, but this is a conceptual slide here, just to stress how new this idea was at the time. So people had understood Church-Turing. So Church-Turing is like, humans are Turing complete or something like that or whatever. And the way they do it is, well, human writes something on paper, moves the paper, writes something, erases. And so they said, okay, well that's Turing machine moves, and so brains are Turing machines, right? But look at McCulloch-Pitts, their argument for why we can be simulated by Turing machines, is that inside the brain are neurons. Those neurons have dynamics. Those dynamics can be simulated by Turing machines, and therefore Turing machines can simulate brains. Okay? It's a different scale, right? Behavior versus internal workings. It was really a fundamental, this is just to point out how powerful this work is. You could even make, new arguments complementary to Church-Turing.
Do you think that's similar to the symbolic versus neurosymbolic, symbolic versus neural AI sort of dichotomy?
Yeah, I think so. In fact, that's what I'm, exactly right. That's exactly right. It's a good way to think about it. In fact, this is how I do think about it, right? So there's McCulloch-Pitts, with the logic gates and stuff. Well, then von Neumann said, great, let's make computers, and then now we have iPhones. But at the same time, they were inspiring this neural computation, which perceptron, and now we get ChatGPT. And I just want to stress one more thing about these amazing people, Pitts and McCulloch, because of course Pitts, they like to switch things around, I guess. But Pitts and McCulloch had another paper shortly after in 1947 called "How Do We Know Universals?" And they were asking, how does a neural network know that this is a piece of chalk and not turns into a dolphin all of a sudden, right? Like it's chalk to you guys, right? When I do this, doesn't turn into a dolphin. And they were applying group theory in a fundamental way to neural networks, and this led to, it was a precursor to Huber-Meisel, and now we see it in what's called invariant or equivariant neural networks. So I do want to, these couple of stories I want to say. So Pitts is, I love Pitts. He's like one of my favorite characters in the history of mathematical neuroscience. And he's the kind of person that thought that there were precious stones in a cliff, got dynamite, blew up the cliff, and found the stones. Okay, that's the kind of person he is. He got hurt in the process. It actually injured him. But that's the kind of person he is. Another example is, so he would go on long hikes, and he'd read books on the hikes, right? And as he'd read the book, he'd rip out a page, rip out a page, rip out a page. And by the time he got to the end of the hike, he had less in his backpack. So he laid less, right? So what kind of person does that? Like that's the kind of person that does, invents these things, right? So a little bit of downside to these kind of people too is, sometimes they crash and burn. Okay, so in particular, he burned his thesis. So he burned his PhD thesis, which was on 3D McCulloch-Pitts networks. He burned it, and he burned it because I can tell you the story afterwards if you want, but because falling out with him and his mentor Wiener. So that's an interesting story. And of course, the sad end of this is that they both died alcoholics months apart. So I think they would have created a lot more excellent stuff. They just stuck to the root beer. Okay, so since then, there's been a lot of mathematics of neuroscience and a lot of principles of neuroscience, and there's tons of progress, tons of really cool books and work on being inspired by neuroscience to understand the mathematics of it and so forth. But when I went from mathematics to neuroscience, I found all of these very interesting and helpful. But I guess I still was confused as to how we were going to make progress when we don't understand pretty much any of the code of the brain. I think that it's a big mystery for us, is what is the brain doing when it spikes? Great, okay, you can make these models and computers and stuff, but what really is going on? And so I was a little disillusioned, but then what I decided to do with my collaborators, like Killing Kopsel, is we said, okay, what can we study? Well, turns out the retina is a little bit of the brain squirted out of your skull. Okay? What I mean by that is it has spiking neurons. It looks like brain. It's a little bit like looking out like this, okay? And so at the same time as it's as complex as the brain, we kind of know what it does, right? It turns this continuous world into spikes through the optic nerve. So it's kind of an encoding algorithm, right? Input comes this continuous world, and out from the optic nerves comes these zeros and ones. And so, okay, let's study retina. And so that's what I'm going to talk to you about is retina mainly, but let's look at sensors. Okay, so this is my claim. I claim that biological sensors are intelligent. That's why I think there should be a working group in this program on sensors. But I also want, von Neumann comes in again. He comes in a lot of places. But I also want to press upon you that John von Neumann even knew that this is the right way to kind of, to do great work forever. Is that math is actually not hard. If you think about how hard brains are, then you think math is easy. Actually, math is like super simple compared to brains, and he already recognized this very early on. Okay, so I'm going to try to convince you in a few slides here that the sensory pipeline is elegant, and I want to do this for a couple reasons. So one is I want to stress upon you that how do you figure something out about the code of the biological system, for instance? Well, trust the constraints, trust the properties, like respect that has to happen in these systems, and then when you make your equivalent model, it should also obey those properties and those constraints. And I would say that's really the way forward. That's the way that McCulloch-Pitts made such great progress, and I think that's also how I looked at the problem with my collaborators. So what are the properties? Okay, so this is a beautiful book, Principles of Neuroscience, amazing book. It talks about how everywhere in the brain is just efficient computation and quantifiably so, and it's a really brilliant book. If you want to know more about what retina does, then please take a look at the book on the left. So let's go through the first one. And just to be clear, let me just mention here. So the claim is that, or the hypothesis is that the sensory pipeline solves many problems at one go. It's not like the best compressor. It's not the best at one particular thing, but the single system itself solves many problems at a very high level. And that's what I want to stress here. So efficient. So turns out when you are in a dark room and I shoot one photon to you, you can detect one photon. One photon. And the reason you can do that is because in the retina, how does it detect photons? There's literally a molecule called channel rhodopsin, which bends when a single photon hits that molecule. So your retina can actually detect single photons. That's amazing. Like, make a sensor that detects one photon, okay? You have to actually have a molecule to interact with the world to be able to do that. So that's part of the efficiency property. There's many other examples of that, but I think being able to detect single photons, in applied physics, that'd be like good thesis, Like if you can make a sensor detect one photon, like Nobel Prize. Okay. Next, so compressive. So it turns, this visual world and movies it's a movie coming at us, right? It's a movie. I mean, it's petabytes, like xenobytes. It's an insane amount of information just coming at you like this. You realize this when you do anything with video processing. You realize that hours of video is going to fill your hard drive pretty quickly. But we're just still sitting here. We're not just like sinking into the ground. So there's a lot of compression going on, and in fact, this This book tries to quantify it with the retina, and we're talking about orders of magnitude of compression, okay? Orders of magnitude smaller the representation coming through the optic nerve. Noise tolerance. So this is another, again, it's ubiquitous in biology, but the example I have comes to mind is, so the retina, you can destroy 30% of your ganglion neurons. Like literally just go hack, 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 destroy them, stare at an eclipse for too long or something, and you will still be able to see fine, no problem. You won't even notice it, okay? It's just when you get to the 31% or 32%, then like catastrophic, right? That's kind of typically how it goes. Progressive, okay, what's progressive mean? So I'm gonna explain progressive in a couple slides like this. So why would you want progressive loading, right? So it turns out, and there are experiments that demonstrate this, the first spikes coming from a sensor, but in particular the optic nerve from the retina, the claim is they convey significant information globally about what's going on in the scene. And why would you want to do that? Is because, here's tiger comes at you. You don't want to go like, oh, there's 3 whiskers here, and you don't care. You want to get the hell out of Dodge. And so that's why these first bits should actually tell you something meaningful, and you shouldn't have to wait around, right, for all of the information, and then you get eaten, right? That's not fun. Okay, so that I mean, maybe it is fun. I don't know, but not to me. Universal. Now this is very recent. This is pretty shocking. If you've never heard of this before, I think you should just have a listen, because this is pretty mind blowing. What do I mean by universal? So what I mean is, and I don't think they can do these experiments anymore. This happened in 2000. They took, I say they, I mean, these neuroscientists took ferrets. Ferrets are these cute little animals, right? Took a cute little ferret. Okay, cute little ferret. What I'm going to do is I'm going to rip out your optic nerve. You don't like that? Too bad, you're a ferret. Okay, so I ripped out the optic nerve, right? And then I pipe it into the auditory cortex of the ferret. Just because, for fun, I guess, right? Just do that. So the ferret is forced to see by hearing its optic nerve. Okay? Guess what happens? It's nuts. The ferret, gets its bearing, is like, okay, and then figures out how to see things. Moves around, performs visual tasks. And the more shocking thing to me was that when you looked at the responses of neurons in this Frankenstein auditory cortex, the neurons started to look like visual cortex neurons. Okay, so here's the picture of that. So this is from their paper. Here's the paper in Nature. It was replicated another time after this, but this was just a stunning paper. So here's a normal, V1 neuron. Here's a normal auditory cortex neuron, and this neuron became a visual cortex neuron. Yeah, that's what happened. So that's what I mean by universal. So I think I'm making okay time. So any questions so far? It's good? Okay. So let's dive into the retina a little bit. So what is the retina? So I'm not going to spend too much time on this slide, but I just want to press upon you how it's a pretty complex thing. It's like that, that ion pump. So what happens is it's pretty interesting. Light comes in to the retina, and then it gets sensed by these channel rhodopsin molecules in the cones and rods, and then all this processing happens in this direction, in the direction of the light, and then once it gets to the leftmost layer there, it gets sent out through the optic nerve through this bundle of 1 million. Think of it like 1 million wires coming out of the back of your eye through that little hole, and that goes to the visual cortex. Here's a little more detail. There's a lot of different kinds of neurons. I'm going to focus on what are called the ganglion neurons. So what are those? Think of ganglion neurons as pixels. Okay, they're trying to encode intensity values of pixels, but they don't quite do that. And let me try to explain how this works. So it turns out that ganglion neurons come in 2 flavors. One is called on, which makes sense. Like if I'm looking at the scene here, there's a lot of bright lights over there, then the on ganglion neuron would go, oh, there's a lot of light. But at the same time, if something's dark in the room, the on neuron doesn't fire, but the off neuron goes crazy. So it's like when things are dark, you're actually having neural activity, even on dark things, and those are called off neurons. So how does it look? And it's even more complicated than that. It doesn't just correlate directly with the intensity value of the light coming in. It has something called a center surround property. So what do I mean by that? So let's pick one ganglion off neuron, okay? So if I show it very bright thing, that's on the far left there, it has two circles. I show it a bright light, it fires like almost nothing, it just barely fires, right? It's not really excited. You put a dark spot right on its, this is called the receptive field. This is where the neuron cares about its input. Think of it as like, image is on top of here. This neuron is looking for this little region around itself. It kind of fires, but it doesn't go crazy, right? What makes it go really crazy is if you do something called a center surround. You put a contrast, okay? So what that means is it's looking not just for absolute intensity, it's looking for like, am I above a mean? Am I above a mean or below a mean? Okay, that's really important. So that's off and on neurons. Another property is that unlike our square grids, it's a hexagonal lattice of neurons. So there's literally hexagons instead of squares, crosses. Another big component to retina is it has different scales of computation. So think of like the smallest pixel is called the parvo. The parvo cell is the smallest receptive field neuron. And there's another neuron which is called a magno, which cares about a bigger region. So think of it has the same properties, except this region will be much bigger that it cares about for the magno versus the parvo would be the smaller area. And something I want to just press upon you that's really important here is that the magno is actually faster than the parvo. The magno has a faster response time than the parvo, which makes no engineering sense. I mean, it cares about a bigger area, has to have more machinery. How can it be faster? It's only faster if the system needed it to be the case and put more energy and resources to make that the case. It's very important. I'll come up with that later. And then another kind of piece of neuroscience is how is the output? What is the output of the neuron? It spikes or not, but what's the code? And right now, a big hypothesis is something called spatiotemporal rate code. What that means is like this over here. So that is the firing rate. Okay, this is going crazy. Okay, right? And it's the counts. It's counts that matter. Okay, they think it's counts. They don't think it doesn't matter if this thing happened a little left or a little right or whatever. They think it's the counts. Okay, that's called spatiotemporal rate code. I don't think anything is the rate code in the brain. I think the brain is too smart to have a rate code, and we can talk about that offline if you'd like. Now to contrast. So that's the retina, but let me just, what is current compression? What do people do in image compression? Because I think it's really important to understand what is the kind of, again, circa 1992, which is still JPEG. You use it on your phone all the time still, right? So it's still pretty current. So what do you do? Compression 101. So you take an image patch. And literally, JPEG operates on 8x8 image patches. And what's the challenge? The challenge is there's a lot of possible images. You have to have a function which takes an arbitrary patch to some code, which means you have to have a function that takes from 2 to the 512 images, a code, right? So what do you do? Well, what you do is you take this DCT basis, which is 2D little pictures. I have 64 of them because I have 64 pixels. So by linear algebra, you can always write a 64-dimensional thing as in terms of a 64 bases, and so you do it, okay? And that will be a perfect coding. There's no error. Then what we do is these higher frequency bases turn out to have pretty small coefficients because of a property of natural images. And so, what if we just truncated that representation by removing the higher frequency components? And it turns out, modulo some technical details. That's what JPEG does. It's JPEG. Make sense? Cut out the high frequencies. And linear algebra is used for expressivity. Okay, so let's go. So let's keep going. JPEG. What does it do? What does it look like? So it's smaller files, high quality, widely adopted. Look, Baboon, great. JPEG Baboon. We're all really excited about JPEG Baboon. What happens if I take 10,000? So out of every 10,000 bits on your computer, 10,000, right? And I flip one of them at random. That's it. So 10,000, flip one. 10,000, flip one. That's it. That's all I do. This is what you get. Okay? You get something that looks like a baboon. Now, if you want to have a modern art, so I have the best generative AI system in the world. Noise a JPEG. okay, the bits of a JPEG, and make a picture, and you'll get this. Okay, there's generative AI for you. Make modern art. Okay, you flip 1 out of every 1,000 bits instead of 10,000, and you get modern art. Okay, that's a modern art. It's fun. Try it fun. Don't do too much because you actually might crash your computer trying to open a 4 terabyte file because you messed up a certain control bit on the JPEG. So don't do that. Here's a little plot that shows what that looks like. So as a function of these bit errors, so how many bit errors do you make? What's the quality? And it just exponentially dies. So JPEG is not a very robust code. It's not noise tolerant. So it's not probably like Retina. Retina's probably not doing JPEG. And what does everybody do? So when you watch your Netflix, I guess people aren't watching as much Netflix anymore, but when you used to watch Netflix all the time and chill, you don't get modern art, right? Okay, you get like Great Baboon or whatever it is you're watching. So that's because for the last 80 years, we have a paradigm to solve that problem, and it's essentially called error correcting code. So you have a source coding, that's compression. You have these red bits which are very fragile, and you can't have that, as I showed you. So you make a bunch of redundant bits, for instance with a Hamming code, and then you pass that down the noise channel, and then you do the reverse of those two things. Okay, so it's a 4-step process really, and this solves the problem, completely solves the problem. However, I would say it's pretty inelegant to me. Right? Look, Hamming code is you take 4 bits, embed them in 3 more, so 7 bits total, to protect 1 only. And if you get 1 more bit, you die. You can't get any recovery. And it just seems like it does the job and it will continue to do the job, but it doesn't seem like what biology would do. Okay, so now I'm going to set you up for biology. So whatever model we had for retina probably wasn't going to be this. This can't be it. So let's start with a puzzle. So how are we going to get in? So I told you this is section 3 now. So I'm making good time, so that's good. So how did we approach this? Again, we approached it by taking really seriously the constraints and the properties of the actual systems. And so I don't know about you, but for me, these 5 bars look lighter than those 5 bars on the left. Do you agree? Who doesn't think that's true? Hopefully, okay, everyone is human in this audience. That's good. So this is called White's illusion. The solution in some sense is that context matters. So notice that these bars are blocking the black, so there's a lot of white here. And these bars are blocking the white, so there's a lot of black there. And so context matters. So your perception is not what each pixel does. Your eye doesn't care about one particular pixel. It cares about it in relation to what's going on in the rest of the image. And so, again, that's a really big constraint. If an engineer you hired did this coding, you would fire them. Immediately get fired, okay, if they made this kind of mistake. So I'm not going to fire Retina. I'm going to respect Retina and say that this was just, it's kind of like, if you want to know this crazy elegant black box, let's find like some error cases which could give insight to witness to some of the complexity inside. This never happens in nature, so no one cares, right? White's, crafted it to make it work. So just because this is last talk of the day, I wanted to give you some more. This is one of my favorite ones too. This is the outside part is a gradient. This is not a gradient. Do you see a gradient? You don't see a gradient? Oh, okay. Nice try. So just to prove it to you, there it is. So it's no gradient, right? It's really quick how that happens, right? Crazy. Okay, more fun. Okay, all these squares, those are the same intensity. This is called Adelson's. A and B are the same. All of those circles are the same color. I don't know if that comes out here very well, but so this is just to show you those are actually the same up there. Yeah. And here's another one for fun. It's called the Hermann grade illusion. I don't know. Do you see kind of some grayish circles kind of appearing and disappearing in the crosses? Right. Okay. So again, I don't think the retina, well, first of all, again, this is controversial. I think this is happening in retina. But I don't think the retina should be fired. I think this is never happening in nature. So as far as the retina cares, like, so what if I do badly on this? It doesn't matter. It is a constraint. So we have to like figure out what is the black box internally doing that has this kind of complexity on these examples. Okay, so I can't So apologies, but until we prepare this paper, I can't give you like a lot of the technical details, but I want to walk you through how I think this idea that we came up with by really considering what retina does, I want to show you some of the properties and see how it might conceivably be related to the biological sensor. And I want to quote this as a cybernetics quote. So this is what we're doing. So at first, I think neuroscience right now is in an empirical sense. Is empirical. I mean, there's a lot of beautiful stuff, and don't get me wrong. There's cool theory. I'm not dismissing it, but I still think I don't think it's mature to the level of physics is. Right? You can't write down an F equals ma yet quite, and so that's where I want to go. I want us to respect the empirical part of this study, which is really hard. Neuroscience is hard. Experimental science is ridiculously hard in neurons. But we learned a lot from it, and I think we should now respect what we've learned, like the things I've showed you, and try to make a logic and mathematics out of it. That's what this quote is saying. Okay, so here's what I'm going to claim. So if you want to see early work already, you can see it in these papers. So this is Sarah Mars and Rametta, others. And we're preparing a paper now. So I claim to you, I mean, that this thing has a lot of properties that are similar to what we see in retina. It's called RMO. So excuse the joke of the name, but the real middle out. So if you've ever seen the show Silicon Valley, the CEO of a new startup invents a compression algorithm called middle out. Okay, so we decided why not? Let's get some, make it fun, call it real middle out. Properties. So let's talk about the properties. So I'm going to focus on just a few of these. So 2 and 3, I think, are really. I mentioned these last time in the sensor slides. So when we walked through how they usually are very orthogonal. To make something compressed, you kind of have to have control bits, right? And those control bits make that code not tolerant to noise, right? Because if you flip a control bit, you're dead, right? So how can it be that this code has this property, these two properties in one go? Let me show you that. So here's a baboon. It's coded with this RMO. Has the same size as the JPEG baboon and the same quality. So they're matched for quality and size. And this is the same amount of error that caused you to get modern art. Here we have errors. There's some errors and stuff, but it's the baboon. I mean, there's no question. That is, I would take that, the right-hand side, like A or B. You're at the eye doctor, A or B. I think B for me. But this points out, so you can actually get a scheme that has a very nice compression and at the same time has a built-in robustness. So how is that even possible? So here's, again, the figure here. This kind of an embarrassing figure to JPEG and PNG. But basically, what I claim is that if you do systematically do this, like you get a linear fall off in quality as a function of noise, which means, think about it, that each bit has about the same perceptual qualia or relevance, right? How do you do that? How is that possible? So here's what it's like, 7-minute abs, and then someone comes out with 6-minute abs, right? So I think like instead of having this 2-step process or 4-step process, you should just have like one move. You should distribute the information evenly. That's the hard part, evenly into the bits. And so when you pass it down a channel, okay, let some of them error, and you don't need to even recover the exact thing. You just need to get it good enough so that you can run away because the tiger's coming at you, right? And so this is what I claim is going on. It's this 2-in-1 process. That's what I think is the, we're inspired by the elegance of biological sensors. It should, it does a similar thing. It codes through a noisy channel by distributing evenly the information into its compressed bits. There's no control bits, and so you don't have to worry about noise. So just to see, people love to see movies. So here's a 1 out of every 2,000 bits I'm flipping. Not something I really want to keep watching. And then here's one out, same amount of error, matched for quality and size. It's watchable, right? I mean, I don't know if you noticed, there looks like there's some little noisy stuff. Yeah.
There's an algorithm that's used on our cell phones where images are sent, and we see them sort of like at low scale, and then they sort of fill in. is this going to work here too? I mean, is there any evidence that that's a biological concept happening at the retina or anything?
Yeah, so I know what you're talking about. So they actually have like, say, 3 different sizes, 3 different scales. So what I'm saying is here, that's 3 different coatings of the one file. So I'll talk about this next. I think what the retina is doing is it's coding one set of bits, and if you want to get a low-res version, you just take a subset of the bits. Whereas they make 3 different coatings. Yeah.
I think that interleaf JPEG does reuse bits. So like it does. I mean, the higher resolution uses also the lower resolution bits.
Okay.
It's not 3.
I thought it
is,
grew. I thought it was layered. Yeah.
Okay, I'm thinking of
that's interleaf JPEG.
the JPEG, like the old JPEG, like 3 scale.
Progressive JPEG.
Progressive JPEG, yeah, which does 3 different Huffman tables and 3 different.
Yeah, no, I think this other.
I'm sure there. I don't want to.
No, I was just wondering. So,
Yeah.
so a couple things. Will this repeat? Can we the same illusions that we see to the eye? Will this algorithm repeat the same sort of visual illusions that we see from our images? Like, right? So the eye looks at these illusions you're showing us and sees little dots. If I use something, I use your algorithm that's modeled after the retina. What properties of the eye will your algorithm?
I'll show you a couple examples, but indeed. So that's what it had to do. So we needed our model to do that, because otherwise it's not retina, right? Again, I'm not claiming we have retina or something. It's just falsifiable, like all these little pieces. So progressive. So just I'm sure when I say conventional here, like engineers are amazingly brilliant and stuff. So I'm sure there's something that is much better than what I'm thinking multi-scale version. But I just want to show you what you can do with this code. So on the left here is imagine like I coded the image, and now I'm just sending you the bits one by one to you, okay? And we're going to see up to 10% as the bits come in. What is the reconstruction of our coding versus, and again, conventional? I'm going to be this is straw person here, okay? Conventional is the bits are coming in, but literally spatially from where they are. So take a look. Okay, so, is this a purple people eater or a flower? I don't know here, but here I know it's not a purple people eater, it's a flower, right? Okay, so so just to see that it really doesn't matter how much, it doesn't, I mean, you can tell already that much of what you need in order to navigate a scene is already contained in the first 10%, right? I mean, it's already all there.
Question?
Yeah.
So I don't remember this work very well, but I remember that people who were doing a lot of Bayesian anthropic prior kinds of image reconstruction would always have these kinds of demonstrations where as you add more and more noise, there's a gradual degradation of the image. It's not all of a sudden it falls apart like those. They're called mandrels, by the way, not baboons.
Magnos and parvos, yeah. They're 2 different kinds of neurons. But...
That was an aside. Those weren't baboons. It's actually called a mandrel.
Oh, okay.
Yeah.
My bad.
Sorry, I was. The main point is that in Bayesian image analysis, there has been work, and I don't remember the details, in which they do have gradual degradation rather than all falling apart. That you do have robustness against noise and so on. And also, especially since Sarah Marzen is involved with this, are there actually Bayesian attributes of your system, or have you looked at those Bayesian systems?
So yeah, with Sarah, we are looking into those things. Yeah. I can't say much more than that, but yeah. But maybe we can talk afterwards, and you can tell me about these references that you're mentioning with the noise tolerance. Yeah. And okay, so I have to show you kind of what it looks like. I mean, this is a code of like black and white, so on and offs, and I claimed you it makes this. This is one of our big promoters is Ali Partovi. This is his son. I did a live demo of taking a picture of his son, and this code on the left is his son. And just to show you that, like inside of it is really there are some interpretable things in there, like you get segmentations out of it and things like that. So this is what this looks like. And then illusions. So the set of illusions that I showed you at the beginning, most of them are called simultaneous contrast illusions. And it turns out that for that class, this model also performs similarly to what psychophysics, what happens in psychophysics. But I think this one's really cool. So here's Hermann grid. And so we coded the Hermann grid with our model, and we didn't see any circles. Like the reconstruction was perfect. So we thought, oh, I guess we have to go back to the drawing board or something, right? Like there's no, it's because it's perfect, like equal. It got exactly the right original grid back. But it turned out, and I showed you this a bit in this code, there's like different kind of scales a bit here. So if you looked in the lower scale bits of the code, there showed up the circles, which might suggest why it's fleeting to you, right? It's not like permanently wrong. It's like this fleeting thing. I mean, I don't know. That was really, really nice to have that work out like that. Yeah.
Are you saying that if you look at the progressive appearance of this image, that you will see the circles at some point?
Yes, that's right. I talked to my lawyer, like a week ago, and I have to be very careful about what I mean, so this is the first time I've ever shown this material. So I would love to show you more, but yeah. So this is really exciting. Like it didn't make an error in the reconstruction. You see the circles in the actual lower scale of the code. And then yeah, please.
You talked about how Huffman coding was expanding the file like by 1.7. What's your expansion rate? Is there one?
Oh, there's no expansion. Yeah, it's just compresses it. The trick is here's the cool thing, is that we don't in error correcting codes. If you have bank receipts and all this stuff, it really matters if a bit was wrong. For us, it doesn't matter if a bit was wrong, and so we don't need to go through this step of error correcting it. It just reconstructs to something a little bit worse. So it's a different idea, right? Instead of like having to get the thing perfect when you get to the other side and error correct, you don't care about the error correction part because the error correction is in the perception of the noised signal, if that makes sense.
But you use real numbers.
No,
It's
it's all binary.
Oh, binary.
Yeah, on off, like up down. Yeah. Yeah, please.
So just following up on that last point, images get copied around a lot. And so if you're not doing error correction and it gets copied, I don't know, goes viral on the internet, gets copied 10,000, 10 million times, what would happen to the image?
No, that's a great question. What would happen is it would degrade linearly until, over here or something, right? And then it would start getting bad, really bad. But it'd be a linear fall off in quality.
That might be a good property. You're interested in intellectual property protection or something.
I was going to say, could you put a signature in there? Could you actually put, exactly. What we want is provenance over images now with deepfakes.
Yeah, I cannot confirm nor deny that that's a great idea. But thank you for your idea.
Just as a reference point, where on the y-axis would we look at the image and think, oh, that's pretty wonky? I mean, is that like at 75%, like 50%?
No, even something like 0.7 is like, okay, it looks...
It starts to look kind of funny.
It's okay. Yeah.
Yeah, okay.
This is really where you start getting modern art and things, but yeah.
So you'd be out like...
It would take a while.
Like 20 or 30% corrupted before, or 20% corrupted before it's...
I mean, you don't want to watch a Netflix movie with 20% of our RMO corruption, but you could, if it's sensitive stuff, you could totally know what's going on.
Are we more sensitive to it because I noticed for the for your image creature
Mandrill, right? Is it? Okay, I gotta fix that in my brain.
For your non-human primate, it was the 0.1% seemed less of a thing than for the video. I don't notice anything there, but when you play the video, you said, oh, like you notice little bits. Is it more? Is the
It's because temporal, you notice things temporally.
decay penalty is bigger?
Yeah, yeah. Yeah, whenever you have moving things, it's a lot, things are noticeable because we like changes in motion and stuff. That's a good question.
Do you know Vijay Balasubramanian? He works a lot with Peter Sterling, for example, of Boston and Sterling. He's a neurobiologist at Penn.
Okay, let me write that down.
So,
What's the last name?
Balasubramanian.
Okay.
He's a fantastic guy. I'm actually collaborating with him on stochastic thermodynamics of popular nets.
Oh, I love that topic.
Oh, if you
Seriously,
want to talk stochastic,
that's a beautiful topic.
My main gig. But in any case, the reason that we're investigating it, this is why I'm asking these things. One of the things that you've not mentioned as a property, a constraint, is that there are, for example, as I understand it, 70 different types of ganglion. There's all these there's a huge number of different kinds of retinal cells.
Yeah.
The obvious question is, what the fuck? Why?
And they're also interacting via amacrines, so they're connected.
He's got a great paper, I sing his praises highly, 2015 in IEEE Spectrum. The more technical version, which may have involved Bill Neumann, Tony Tushby, I think, was neural computation before that. But basically, he relates it to something that you've actually not mentioned yet, even though Laughlin and Sterling make a big point of it, which is thermodynamic efficiency. He says that he makes the case that by having different channel capacities of all these different neurons and reverse multiplexing, you can get the same information down those wires with far less energetic cost. And that's actually front and center with Laughlin and Sterling and recent work by Levy and Calvert in PNAS, all about thermodynamic costs and what the implications are for architecture of the brain.
Mm-hmm.
With Sarah on this paper, I have to ask, and if you say can't talk, I understand, but that's a constraint that you haven't said anything about. One of the major constraints of the brain is so little energy burn.
Yeah.
That is the big, most important distinguisher, frankly, compared to everything else that we built, is the amount of energy it requires.
Yeah, so I love that point. It's a great point, is that so each pixel or whatever in the retina has two wires, right? The on wire and the off wire. And so if you send something on, a bright thing, you're sending 1 unit of energy, which is very costly. It's very expensive to send it. And if you have something that's an off neuron firing, it's also 1 unit. But they never both fire, okay? But what about if you send no information, no on or offs down the wire? Turns out that's signal. And I can't get into it, but that's basically one of the remarkable advantages of this paradigm, is that by sending nothing, you actually send almost everything. It's a deep thing. I'm happy to talk to you about that offline, but this is something that Sarah's also very interested in, yeah. That's a good point. So the last thing I'll close with, so I'm almost done, is thank you for all your questions. I want to talk about how this relates to experimental neuroscience, because I think that this is very recent that we looked at this, and I think it's really exciting. Okay, so this is work from the Chicholnitsky lab that he does a lot of retinal neuroscience. So what they do, imagine I have a retina, and I'm showing a bunch of images, and the eye moves around. It's called microsaccades. It turns out if I look at something and don't move my eye, it disappears. Okay, so your eye actually has to move around a bit. They're called saccades. So imagine I'm showing a retina, a bunch of images, and I'm saccading around. And now let's pick 2 neurons. These are 2 different neurons, many trials, same protocol. So I do the same protocol every time. This is 1 neuron over, and each row is a trial. So it's pretty stereotyped, right? It looks like this neuron, given the same input, basically performs the same calculation. And here's another neuron. Also, just get a sense, these are natural images. It's very important, these are natural images. This is kind of how the firing looks like of these neurons. Here's some more. And however, if you give white noise to the neuron as the protocol, this is how the firing looks of the neuron. Does that make sense? Like this is over trials, same protocol. Excuse me. It turns out that what has been really hard for everyone in the field of retinal neuroscience, they can predict with a GLM, they can predict, given the input image, they can predict what the retina would do when it's white noise. Okay, so they can predict these spikes here. They can't predict these. They can't predict from their GLMs on general linear models, given an image patch. They cannot predict the natural scenes. So what's going on there? So it must be somehow that there's a non-local kind of system, right? Remember, how is it possible that I can compress an image evenly into bits? It's because each bit saw the rest of the image in order to compute what it is. It's not this local little bit. It actually cared about what everything else happened, everybody else talked to everybody, did everything, and then now that bit has a decision on or off. And what that does is it makes it a non-local computation, right? It cares about what's going on in this scene. And so you can imagine if I have an image moving around, there's some big scale features which if I moved over enough, all of a sudden they change kind of what should happen locally. And so here, what I'm going to show you now, so this is my grand finale here. I'm going to show you, I'm going to do the same protocol, but I'm going to take one of my bits, okay? And think of it as like an on neuron or something. And I'm going to move the thing around just like they did here. Same protocol. And I hope that the things will look like this for natural scenes and like this for white noise, right? Make sense? Okay, so indeed they do. So this is my, my model neuron, and I'm just watching what you're doing to the model neuron as you move natural images around. And this is the phenomenology. Of course, I didn't match, you can't, this is just the phenomenology looks the same, but you can see that there's definitely a pretty significant difference between natural images and white noise under the model, right? And this got us very excited because again, it's another falsification. Like if our model didn't do something like this, then it's probably wrong. So it's good. It's good that it looks like this. Remember, this is how they looked with real neurons. This is natural scenes, and this is white noise. Coming back to the model. This is natural images and white noise. And in fact, just to finish off here, I'm almost done. What about uniform noise? I think they should do a lot more statistical classes of input to neurons. I think that's really valuable. And so what do you think? So instead of Gaussian noise, so the white noise is these little independent Gaussians, what if I did uniform noise, right? So I picked uniformly an intensity value, uniformly. What do you think would happen? Do you think it'd look more like this, like that? I don't know. So turns out, it looks like this. It sharpens somehow. So here's a prediction. I mean, they could just do the experiment. Maybe they already have the data. If people don't like uniform noise, right? Maybe why is because they're too predictable or something. I don't know. But makes a prediction. Okay. So thank you. These are my my funding agencies from long ago, and these are some recent VCs that have been really interested in this work, and so thank you for your time.